Hello, I'm Dr. Aruna Saman, and in this video, we are going to discuss asexual reproduction in lower organisms and in angiosperms. As we all know, reproduction is one of the features of living organisms. It is an ability of living organisms to produce another living organism of its own kind. What is the significance of reproduction? It leads to continuation of species and it maintains the continuity of life on this earth. The reproduction is mainly of two types, asexual or apomixis and sexual reproduction or amphimixis. What are the features of asexual reproduction? It is uniparental. Only a single parent takes part in the process of reproduction. It does not involve meiosis or reductional type of division. There is no fusion of gametes or fertilization, and hence there is no mixing of genetic material. That is why it is called apomixis. Apo means without, mixing of genetic material, and hence the progeny produced is identical and it is called clone. Types of asexual reproduction in lower organisms. In Spirogyra, we come across fragmentation. In yeast, we find budding as a method of asexual reproduction. In Chlamydomonas, the spore formation, the zoospores, the motile spores, and amoeba, it shows binary fission. In sponge, it is gemule formation. Let us see fragmentation as is observed in Spirogyra. The filament or the multicellular filament, it breaks into many pieces due to some reason. These are the different fragments produced, and each fragment can grow into the fully developed filament. Thus, from a single original filament of Spirogyra, number of filaments are produced by the process of fragmentation. The budding or the example is yeast. It's a very common in this unicellular yeast organism. One or more outgrowths called buds are produced, and these buds when separate from the original cell, they develop into new individual yeast cells. It all happens in favorable condition. Once again we can see here a bud has started developing on the parent cell. Then the nucleus of the parent cell divides and produces two nuclei. One of the nuclei enters the bud, and then the bud becomes a separate daughter cell. Within a very short time period, the chain of new cells are produced from the single original cell by the process of budding. Zoospore formation is observed in Chlamydomonas. The matured independent haploid cell of Chlamydomonas withdraws its flagella and divides twice by mitosis. As a result, 4 cells are formed. These daughter cells later on develop flagellae and cell wall. These are released from the parent cell. Since they can move, they are motile and hence they are called zoospores. These zoospores then develop independently into mature haploid cells. Other methods of asexual reproduction in lower organisms are conidia formation observed in Penicillium. Conidia are also spores, but they are non-motile spores. Binary fission observed in amoeba and gemule formation in sponge. Types of asexual reproduction in angiosperms, which we call higher plants. Vegetative reproduction is the most common method of asexual reproduction in angiosperms. When the plant propagates through its vegetative parts, which are not meant for reproduction, such as root, stem, leaves, it is called vegetative propagation. These vegetative parts act as propagules, and to act as propagules, they must have growing points called buds on them. Sometimes they may contain reserved food material also. Natural vegetative reproduction in angiosperms. It can be by root, as is observed here. The underground root stores food. Or it can be with the help of leaves, where buds are produced on the margin of the leaves. Or it can be through stem, which are underground or subaerial or aerial. Let us see the different types of vegetative reproduction in angiosperms, which are divided into two categories, natural and artificial. The natural methods of vegetative reproduction are by roots, by stem and by leaf. By root, as we have seen earlier, the tuberous roots as in sweet potato, or it can be a non-fleshy roots as is observed in the muraya plant or guava plant. By stem, it can be underground or subaerial or aerial. The underground types of stem are bulb as is observed in onion, or it can be cork as in amorphophilus, stem tuber in potato and rhizome in ginger. The subaerial methods of vegetative propagation are runner as is observed in grass like cynodon, stolon as is observed in strawberries, offset in pistia and sucker in chrysanthemum, mint, banana, etc. The aerial method of vegetative reproduction is observed in Dioscorea in the form of bulbils. In leaves, we find the bud production in plants like Bryophyllum, Kalanchoe and Begonia. The artificial methods. Horticulturists and agriculturists have developed several methods and techniques for the propagation of varieties of economically important plants. And these are the artificial methods of vegetative propagation. There are three main techniques: cutting, grafting, and tissue culture. Let us first see the cutting. Small pieces of vegetative parts of a plant are used for propagation. These parts should have one or more buds on them. As is seen here, in rose, it is stem cutting, so also in bougainvillea. In sansevieria, it is leaf cutting, whereas in blackberry, it is root cutting. In grafting type of artificial vegetative propagation, as is done in apple, pear, rose, etc., parts of two plants are joined in such a way that they grow as one plant. In this method, the rooted plant is stalk, whereas the other part of the plant is scion, which has all desired characters and also has one or more buds on it. This scion is joined onto the stalk. The buds of the scion are allowed to grow further, but there is no bud growth on the stalk plant. The bud grafting is also done where bud acts as scion and on a stalk plant a T-shaped cut is given. The bud, which is now scion, is inserted in this T-shaped cut. A tape is used so that the bud remains in contact with the stalk, and in due course of time the tissues of the two merge and scion starts growing on the stalk. This is bud grafting. Tissue culture. It is a laboratory method of vegetative propagation where a small amount of tissue is taken from parent plant. It is called explant. This tissue is grown on agar medium which contains all nutrients and plant growth hormones. In due course of time, that tissue divides and redivides and forms an undifferentiated mass called callus, and with the help of different growth hormones in correct proportion, organogenesis takes place and new plantlets develop. These plantlets are first grown into a compost, and then these baby plants are transferred in the fields. This way, by tissue culture, from small amount of tissue, number of new plants, which are all clones, are produced. Micropropagation. It is also a method of tissue culture, but the explant is shoot apex. And this shoot apex is allowed to grow further and produce number of new buds, and they are separated to produce new plants which are clones. This is done in a very short time period. The technique is applied to commercial vegetatively propagated plants such as banana. What is the significance of vegetative propagation? It is very easy and cheaper method of multiplication. Genetically identical organisms get formed. It's a very fast method. It is useful in plants that do not produce viable seeds such as banana. It increases in yield by grafting or layering method, and desired characters can be preserved. They can be combined by grafting like methods. We have come to the end of our discussion on asexual reproduction in lower organisms and in angiosperms. Thank you.